Hello, and welcome to this guide on interpreting a clopidogrel pharmacogenetic test report. During this presentation, I will begin with a short section on pharmacogenetics and common genetic terminology. As further background, I will briefly look at the relationship between CYP2C19 genotype, CYP2C19 metabolizer status, and the impact on clopidogrel prescribing. The main objective of this presentation will be to examine and interpret an example test report and use this information to help guide personalized prescribing of clopidogrel. Genetics is the study of an organism's genetic makeup or genotype and how it affects their physical characteristics. In pharmacogenetics, the phenotype of interest is the pharmacology of a drug and how it is affected by an individual's genotype. This term is often used interchangeably with pharmacogenomics. It is also important to understand the two main branches of pharmacology, pharmacodynamics and pharmacokinetics. Pharmacodynamics is the study of the impact of a drug on the organism, while pharmacokinetics looks at how the organism processes a drug. Genes are segments of an organism's DNA that contain code to produce a protein. In the context of pharmacogenetics, these proteins may be involved in pharmacodynamics, for example drug receptor proteins, or pharmacokinetics, for example protein enzymes involved in metabolism of drugs. Variants or mutations are changes to DNA that can affect the protein product, changing the function of the protein, increasing its activity, or making it less active. A homozygous variant means both alleles are the same, and heterozygous means a different allele has been inherited from each parent. A single nucleotide variant, or SNV, is a single change at a point in a DNA sequence from the letters A, C, G, and T. The cytochrome P450 or CYP450 enzyme family are a group of liver enzymes responsible for metabolizing around 90% of drugs. CYP2C19 is one of the members of the CYP450 family and metabolizes a range of commonly prescribed drugs. As with other CYP450 enzymes, there is a large amount of genetic variation in CYP2C19 and there are 39 known variants. This diagram shows the different CYP2C19 metabolizer groups and how their level of CYP2C19 enzyme activity on the y-axis relates to the speed of metabolism of drugs metabolized by CYP2C19 on the x-axis. Those with lowest levels of CYP2C19 activity, the poor metabolizers, are shown on the left, and as you move towards the right, an increasing level of enzyme activity is associated with an increased speed of metabolism. These metabolizer subgroups are usually determined from an individual's genetic profile, but it is worth noting that environmental factors such as liver disease or drug interactions also play a role in level of CYP2C19 activity. The Human Genome Variation Society, or HGVS, provides recommendations on a standardized method for reporting genetic variants. CYP2C19 here denotes the gene. C indicates that the reference DNA is the coding region of the gene, while -806 shows the relative position to the reference coding region. At that position, a T is present in the sequence, instead of the C seen in the reference sequence. While this method is used for naming SNVs that are important in applications such as rare disease diagnosis, the lengthy naming system does not work as well for pharmacogenetics. This is because in pharmacogenetics, in each allele, often multiple SNVs are inherited together as a haplotype. To make the process more straightforward, a so-called star allele naming system for pharmacogenetics was introduced by a Dutch pharmacologist. Instead of the HGVS nomenclature shown in the last slide, this allele can be described as CYP2C19 star 17. Star 1 usually denotes the wild-type allele. Wild-type is a term to describe the most common or normal version of an allele. In order to determine an individual's metabolizer phenotype, the combination of star alleles inherited from each parent, also known as diplotype, is needed. For example, if two wild-type alleles are inherited, their diplotype would be CYP2C19 star 1 star 1. This table shows how some of the different star alleles impact an amino acid in the chain that makes up the CYP2C19 protein. In the star 1 wild-type allele, enzyme activity is as normal. In CYP2C19 star 2 and star 3 alleles, the protein produced has no function, while the star 17 allele produces a protein with increased function. Note that some of these are more prevalent in certain ethnic groups, illustrating how CYP2C19, like other CYP450 enzymes, varies across geographical regions, both within and between biogeographical populations. By combining the effect that each allele has on the protein produced, we combine this to determine the overall metabolizer status. For instance, 2 star 17 alleles both have increased function, and that combination leads to an ultra-rapid metabolizer status that is seen in the bottom left. Using the alleles star 1, star 2, star 3 and star 17, this table outlines the possible combinations that can lead to each of the metabolizer phenotypes. One of the drugs metabolized by CYP2C19 is clopidogrel. This is an antiplatelet commonly prescribed to decrease the risk of cardiovascular and cerebrovascular events, among other indications. As a prodrug, it needs to be metabolized by CYP2C19 to the active form to take effect. In individuals with reduced CYP2C19 metabolizer status, formation of the active form is impaired, which can reduce the effectiveness of the drug and lead to a greater risk of cardiovascular and cerebrovascular events. Having looked at an overview of CYP2C19 and clopidogrel, we will now look at an example test report. In a clinical scenario, a pharmacogenetic report for prescribing clopidogrel may look like this. Note that this is a mock report and does not relate to a real doctor or patient. In the following slides, I will examine each section in more detail. At the top are the details of the testing centre. North Thames Genomic Laboratory Hub is the name of the lab that has performed the test and generated the report. It is based at Great Ormond Street Hospital in London. In the middle are the details of the fictional clinician Dr. Yadev, a stroke consultant. This is a test that has used a blood sample collected on 30th of May 2024, and the report has been issued on the 6th of June 2024. At the bottom are the details of the fictional patient Akiko Suzuki, along with the date of birth, NHS number and hospital number. In a report, these details should be checked to ensure they match the patient. The purple box with blue text at the top demonstrates that this is a pharmacogenetic report for variants in CYP2C19. In the black box, the reason for testing is that the clinician wishes to obtain the CYP2C19 genotype. The results summary shows that the individual has a pair of star 2 alleles and is designated as a CYP2C19 per metabolizer. The result and interpretation states that 3 variants are tested for. Star 2 and star 3 are both loss-of-function variants. Star 17 is a gain-of-function variant, or in the context of pharmacogenetics, is an increased function variant, as gain-of-function can sometimes indicate a variant with a completely new biological function. Note that star 1, the wild-type allele, is not tested for, but is only inferred in an allele if star 2, star 3, or star 17 are not detected. Below this, the report states that Akiko Suzuki is homozygous for star 2, meaning that both alleles are the same. This predicts Akiko to be a poor metabolizer. This section finishes with a cautionary note that the CYP2C19 metabolizer status may affect multiple medicines. These include selective serotonin reuptake inhibitors, or SSRIs, and proton pump inhibitors, PPIs. It refers to the Clinical Pharmacogenetics Implementation Consortium, or CPIC, guidelines for prescribing recommendations and advises to discuss queries with a local pharmacist. The report finishes with details of test methodology, the reference sequence, limitations of the test, regulation, and accreditations. Having read the test reports, the next step is to consider how CYP2C19 poor metabolizer status can impact prescribing of clopidogrel. While the report refers to CPIC guidelines on the CPIC website, it is worth noting that it may be easier to check the recommendations from CPIC guidelines using the website www.pharmgkb.org. On that website, other international guidelines are also available. It is also important to consider drug regulation, which can be found in the Summary of Product Characteristics, or SMPCs, at www.medicines.org.uk. In this scenario, I would avoid prescribing clopidogrel following CPIC guidelines. In the case of stroke, this report shows that a stroke consultant ordered the test. It advises use of an alternative P2Y12 inhibitor instead of clopidogrel. Different prescribing recommendations are available for cardiovascular indications. Also remember usual clinical considerations when prescribing, such as age, comorbidities, clinical parameters, and drug interactions. Thank you for listening to this presentation. For further study, additional reports can be found on the North Thames Genomic Service website. If you have time, please attempt to interpret these reports in the context of clopidogrel prescribing for stroke.